coming to the 10th topic, which is metabolic basis for living.
Okay.
So here we are going to see various processes, metabolic pathways, like some molecules, okay, from complex structure, they become simple structure.
By itself. Okay.
And some molecules from simple structure, they become complex structures.
By itself. Okay.
By various metabolic pathways. Various metabolic enzymes are acting here, okay? Various metabolic pathways and everything. So here, when a simple structure develops into a complex structure, it is known as anabolic pathway.
Anabolic pathway.
Yes, because it is creating from simple, it is going to complex.
Okay.
So for example, if you take acetic acid, it becomes cholesterol. Okay, so it is anabolic pathway.
So cholesterol become acetic acid?
Yeah, that's what. When simple structure when complex structure forms simple structure. From complex it is becoming simple structure.
Yeah.
So from cholesterol it is becoming acetic acid. So it is catabolic pathway. Okay, so example of this catabolic pathway is glucose. Okay, glucose which becomes lactic acid in muscles. Okay. So this is simple structure from more complex. Okay. So this anabolic pathway, since it is creating more complex structure from simple, it needs energy, it requires energy. Whereas catabolic pathway, it breaks down the complex structure, it breaks down the complex structure and it breaks down the bonds and it becomes so simple.
It loses its energy.
Yeah, it loses it and it gives out energy. Okay.
The gained energy is it gives out.
Yeah, it gives out energy. It can be any other examples. Okay. So here, energy, these released energy are stored in a particular chemical bonds. Okay. So after storing into chemical bonds, whenever the organism needs energy, it breaks down the bonds and it uses up the energy. Okay. So most important energy currency is bond energy in chemical called ATP, which is known as adenosine triphosphate. We have already discussed about this.
Adenosine triphosphate.
Yes. So this is the bond formed by energy, and whenever the organism needs energy, it breaks this bond and it obtains energy. Okay. So there is a study called bioenergetics. Okay. So where here the strategies involved to derive this ATP, to store this ATP, how it is stored, the ATP, how it is stored, and how this ATP is converted into energy. So there are various mechanisms where we study in bioenergetics.
Okay.
So there is a study of these energy bonds. So with this, the metabolic basis of living topic ends. So next we are going to the next topic, which is the living state. Now we are going to see the 11th topic, which is the living state.
Okay.
So here the living state, so there are generally biomolecules, they are present in various amounts of concentration. Biomolecules, for example, in blood the glucose level is 4.5 to 5 millimoles, and in hormones are present in nanograms, which are 10 to the power minus 9 grams. And so these are present in various amount and varying range in every organism. Okay. So here the living state is nothing but so system is not in equilibrium. It continuously performs some functions, not in equilibrium. So in physics you will be saying that if system is at equilibrium, it does not perform function. It does not perform any work. So our body performs work. So our body is not equilibrium. And so living state is not equilibrium. Okay. And it prevents falling into equilibrium. So to prevent, we have to not go into equilibrium. If we go into equilibrium, we cannot perform any work. Okay. So it should also prevent falling us into equilibrium. So to prevent falling into equilibrium, that is some energy needed, right? Input. You have to do that one, this one, and constantly it has to be maintained in not equilibrium. Stabilize is equilibrium. It should not be. So here there is a need of some energy input. So this energy is obtained by metabolism. So metabolism is where catabolic and anabolic take place.
In the last topic.
Yes.
Okay.
So metabolism constantly provides energy, so therefore by getting energy it prevents us to fall into equilibrium, so therefore we will be in non-equilibrium and capable of performing work. Okay. So here, since this living state and metabolism are interconnected, so they have, if living state is not there, metabolism is not there. If metabolism is not there, living state is not there.
Both are
interdependent.
Okay.
So that is what is the living state. Okay. So we are not in equilibrium. Okay. So that's what we do.
But we need equilibrium.
Why we need equilibrium?
We don't.
We know it. We have to not be in equilibrium. Then only we can perform functions. Okay. So now we are going to discuss about the 12th topic, which is the enzymes.
Okay.
So here almost all enzymes are proteins.
Proteinaceous. Okay.
They have amino acids in them. Okay. And some nucleic acids sometimes, like DNA or RNA, some nucleic acids sometimes, they act as enzymes.
Okay.
For example, ribozymes. Okay. So these enzymes to catalyze reaction, these enzymes are nothing but catalysts. So to catalyze a reaction, to make the reaction faster, to happen faster, they have a pocket-like structure in their secondary structure. Okay. In their tertiary structure called active sites. It is a pocket-like where the substrate comes and fixes and it is converted to products. Okay. So there are inorganic catalysts and organic enzymes. Okay. Inorganic catalysts work at high temperature and pressure.
Okay.
Whereas enzymes damage at high temperature. But if you isolate enzymes from organisms that are living at extremely high temperatures, so they have thermal stability in them, so they can work at a little bit high temperature. Okay, but normally all the enzymes are damaged at very high temperature. They are denatured. Okay. So this is about enzymes. So we will be seeing in detail about enzymes, how it structures and what are the process to form products is here, and we will be seeing in detail in enzymes. Okay. Now we are going to see chemical reactions under enzymes. Okay. So here. Amount of product formed per unit time. Okay. Amount of product formed. Okay. Per unit time. Per unit time is generally taken as second. Okay.
Okay.
So it is known as rate.
Okay.
So rate is equal to amount of product formed per time. So total number of products per total number of time. Okay. So this is rate. So it is highly influenced by temperature. Okay. So when rate doubles or decreases by half for every 10-degree change in temperature. So if any 10 degree increases or decreases, the rate changes, it doubles or decreases by half. Okay. It either multiplies into 2 or it divides by 2. Okay. So here, for example, if you take CO2 plus water to give hydrogen carbonate, H2CO3. Here, in the absence of enzyme, this reaction takes place about for 200 molecules per hour. For 1 hour, 200 molecules of H2CO3 are produced. Okay. The same with enzyme carbonic anhydrase, which catalyzes this reaction. Carbonic anhydrase, this is the enzyme used here. So if this enzyme used here, the reaction completes and the number of products formed are 600,000 molecules per second. Okay. So it generally increases 1 million times these enzymes. Okay, it reduces the time period. So in different conditions, different products are formed. It is dependent on conditions. Okay. For example, in muscles, in human muscles, skeletal muscles, under anaerobic respiration, under anaerobic times, the energy is converted into lactic acid. Okay, glycogen is converted into lactic acid. So at the same time, in aerobic condition, it is converted into pyruvic acid. So it depends on conditions, whether anaerobic or aerobic. So it depends on different conditions. Okay, can you understand? So this is which comes under chemical reaction. Okay. So next we will see about how enzymes perform at so how enzymes bring about these chemical reactions. Okay, we are going to see in the next chapter.
How enzymes bring about
Chemical reactions. So now we are going to see the second subtopic under enzymes, which are high rates of chemical composition. Enzymes at high rates of chemical composition. Okay, how do enzymes bring about this one? So here to explain this, there is a diagram here.
Okay.
So this is potential energy,
Okay.
and this one is progress of reaction.
Okay.
So this is activation energy with enzyme, and this one is activation energy without enzyme.
Okay.
Okay. So here energy is nothing but the energy needed by the molecules. Okay, activation energy, the energy needed by the molecules to form products. Okay. So without enzyme, it means more number of energy and the more energy and more potential energy. So with enzyme, it it comes here, right? So it means less energy, so less potential energy. So they form products easily. This is substrate and this is products. Okay, they form products very easily. So what is the mechanism? What is the nature of enzyme action here? This is the third subtopic actually. So we are going in detail about it. Okay. So here, this will be mentioned in all subtopics. Even in this subtopic, this is mentioned here. But I have they have also given this separately. Okay. So we will just see separately one. So E is nothing but enzyme. S is nothing but substrate. Okay. So we have already seen that an enzyme, if you imagine this is an enzyme, it will be having some active site. So this is known as active site, some position in that enzyme. Okay. So with this enzyme, substrate comes and attaches here. Okay, so it comes and it fixes here. It is known as
Fills the gap.
Yeah, it fills the gap and it is known as E plus S. Okay. So when enzyme and substrate, yeah, this one is enzyme. Okay. So enzyme substrate is nothing but the substance which has to be changed into products. Okay. So ES is nothing but the enzyme and is changing the substrate. The process is going on to become product. Okay. So it is attached with this. Okay. The process is going on to form products. So there are 4 stages here. So this is the first stage, second stage, and the third stage is nothing but if this is the enzyme. Now it has formed the products. It is within this is the active site node to form products. Okay. So here the substrate is converted into products.
EP.
Yes, EP. So now the fourth process is the enzyme releases the product.
Why does it release a product?
Yeah, the enzyme, it just makes the reaction faster. It
Transports it. Converts it.
Yeah, it converts the products faster. Okay, so it takes a substrate, it converts into products and it transports it faster.
Fraction of second.
Yes, within a fraction of a second. Okay, so it releases the products from its active site. This is the mechanism taking over next enzyme substrate mechanism. This is known as the enzyme substrate mechanism. So E plus S, ES, EP and E plus P. So first enzyme comes and attaches and the enzyme starts the substrate to process, and after processing it forms products which are fixed with the enzyme, and after some time it releases. So this is the mechanism, and this is an example how fast the enzyme. So this one have lower activation energy. Lower activation energy with the use of enzyme. Okay, so it reduces the activation energy. So with the less amount of energy, it can form products. So if it has more activation energy, it needs to get more energy to form products. That is the concept here. Okay. So now we go to the next topic, which is factors affecting the enzymatic activity. Okay. what are the factors which affect enzyme will be seen. So now we are going to see the factors affecting the enzymic activity. So here, what are the factors affecting the enzymic activity are temperature, pH. So they need an optimum temperature and optimum pH. Okay. So optimum temperature is nothing but the temperature which is not too smaller range or too bigger range. It should be
neutral, near neutral.
It should be, yes. It should be within the range. Okay. It should not be too small or too large. Okay. So this is known as optimum temperature. Okay. So if it is too lesser, the temperature is too low also, the enzyme will not work. And if the temperature is too high also, the enzyme will not work. At the current temperature only the enzyme works at this point. So if it is too high, enzyme does not work. If it is too low, enzyme does not work. The same condition is for pH also. Okay, if it is too acidic, the enzyme cannot work, and if it is too neutral, the enzyme cannot work. It should be the medium of neutral and little bit acidic and little bit basic. So it should be in that condition. Then only the enzyme activity will be useful. Okay, so it can produce a large number of products. So now substrate concentration, it is also a factor affecting enzyme activity. So first, when the substrate concentration are when there are more substrates, okay, the enzymic activity first it enhances, first it reaches the maximum point. And while the continuation of the process, when the substrate concentration is still more high, then the enzymic activity will be less, because there will be no enzymes to obtain that substrate in its active site. The enzymes will be over, then the substrates will be remaining. Okay. So substrate concentration, if it is high first, the enzymic activity is high at the starting. It reaches the maximum velocity. Okay. So now inhibitor. So there are some inhibitors in enzyme which inhibits the enzymic activity. Okay. So how it inhibits the enzymic activity? Actually the enzyme is like this. So the attaching here are substrates. So instead of these substrates, other than the substrates, other molecules, okay, which have the same molecular weight of the substrate, same structure of the substrate and all the characteristics of the substrate, they attach to this active site. Okay, and therefore they inhibit this enzyme from working and giving out products. So the substrate will not give products. So it is known as inhibitor and the process is known as inhibition. Okay. So this closely resembles the substrate, this one, the other molecule which closely resembles the substrate in molecular weight, structure and inhibits the activity of the enzyme. So this type of inhibition is known as competitive inhibition or competitive inhibitor. The enzyme the molecule which is inhibiting is known as competitive inhibitor. Okay. So example of this is the inhibitor is melanoate. It closely resembles the substrate succinate. Okay. It resembles the succinate. Melanoate resembles the succinate and inhibits the formation of succinate dehydrogenase. Okay. So this is these are the factors affecting enzymic activity. So these factors, the enzymic activity are affected: pH, temperature, substrate concentration and inhibition. Okay. So next we will see about the next subtopic is classification and nomenclature of enzymes. So now we are going to see classification and nomenclature of enzymes. Okay. So here the enzymes are classified into 6 classes and each classes have 4 to 13 subclasses according to the nature of the enzymes. Okay. So here we are going to discuss some of the classes here, some of the nomenclature and classification here. So enzymes of oxidoreductase or dehydrogenases, they generally do the mechanism of like oxidoreductase, they oxidize as well as they reduce. Like if S is reduced here plus S dash oxidize here, so it gives S is nothing but a substrate. You just keep it as an example. So here it is S is reduced, but here it is oxidized. Here S dash is oxidized, but here S dash is reduced. So this is the oxidoreductase or dehydrogenase. Okay. So these enzymes, they reduce as well as they oxidize the substances. Okay. So the next one we are going to see about transferases. So here what does these transferases do? If any substrates like S linked with G plus S dash, okay, what they do is they just transfer this T from S to S dash. So it results in S plus S dash which is linked to G. So like this they transfer molecules from molecules attached to other molecules to some other molecules. Okay. So hydrolases are nothing but which breaks bond through supplement of water like that. So these enzymes it hydrolases and breaks bonds. So then lysases. So what does these lysases do? If two carbons attached with some other substrates, these lysases joins the other substrates which are attached to the two carbons. So the two substrates are joined and they are separated from these two carbons. So these two carbons get double bonded carbon structure. Okay. So this is about lysases. And then we are going to isomerases. So what does these isomerases are nothing but they do interconversion of optical geometry and positional geometry, that is positional isomers. Geometrical and positional isomers, they convert the position of the compounds. Where are the carbon aberration? They will be turning off the position. Like we see in chemistry, you will be seeing optical rotation and everything. So this isomerases comes there. It plays a role in the positional and optical geometrical change. Okay. So and then ligases. So what does these ligases do? They just link two compounds or two substances. For example, carbon and oxygen are separate compounds. They link these together into carbon monoxide. Like that they do. So these are ligases to join the separate compounds. Okay. So we have discussed about some classification of enzymes, but there are various classification of enzymes. So next we go to cofactors under enzymes. Okay. We discuss about cofactors. So do you know what are cofactors?
Factors that help for moving process.
Yes. So these cofactors are non-protein, which are bounded to enzymes. Okay. Which are bounded to enzymes to make them active. So these cofactors are bounded to enzymes. If these cofactors are bounded to enzyme, then the enzyme will be more active. Okay. So the protein portion here is known as apoenzyme, which is nothing but the enzyme which is known as apoenzyme, to which the cofactor is attached. So these cofactors are of three kinds: prosthetic groups, coenzymes, and metabiomes. Okay. So first we are going to discuss about prosthetic groups. So prosthetic groups are nothing but organic compounds. So these are a kind of cofactor which is bounded to the apoenzyme. So they are tightly bounded to the apoenzyme and increases their potential. So example, peroxidase and catalase. Okay, these both are catalysts to catalyze hydrogen peroxide to break them into hydrogen and oxygen. Okay, so here heme is used as prosthetic group. Okay, then the second one is coenzymes. So coenzymes are organic compounds. So they are not tightly bound as prosthetic groups. They are less tightly bound. Okay, so they are transiently with apoenzyme. They are just attached but not tightly bound. Okay. So these are generally vitamins. These are vitamins. Example NAD. NAD is nothing but nicotinamide adenine dinucleotide. Okay, and NADP contain vitamin in them. Okay, vitamin niacin in them. It is very important. Vitamin niacin in them. Okay, so they increases the potential of apoenzyme. And the third one is metabiomes. So metabiomes are inorganic. So since till now we have seen prosthetic groups and coenzymes which are organic, but these metabiomes are inorganic. And these are coordinated bonds with side chains at the active site. Okay, do you know what is active site? We have discussed active site are present on the enzymes. So when the substrates are attached, these metal ions form, they coordinate bond between the enzyme and the substrate. So that is what its function. So they coordinated bonds with side chains at active site. Okay. So if you, for example, zinc is a cofactor for carboxypeptidase.
Okay.
So zinc is a cofactor for carboxypeptidase. Okay. So when these cofactors are removed, the catalytic activity of the enzyme will be lost. The enzyme will not be catalytic. Okay. So this the chapter ends cofactors. So if you like this video, please subscribe and please click the bell icon which is nearby. If you have any doubts, please leave them in the comment section.
Pritika and Anil signing off.
Thank you.